Dear young learners, today we are going to discuss about glycolysis preparatory phase 1. So let us discuss about glycolysis. Glycolysis, it is a very simple process. When you consume some amount of food, any different variety of food, anything if you consume, it will be mixed. If you crush well and mix with saliva, and it will travel along with the esophageal layer, and there is the entry of small intestine, large intestine, and finally it will be in villi. Through villi, the micro macronutrient it will absorb, and finally it will secreted in the blood tubes. You can very well see the blood tubes. There are the micro macro foodstuff. Finally, the glucose can be generated, and the glucose can be transferred from blood tubes to working tissue. For this process, there are nearly 18 stages, 18 coenzyme it will involve for transfer from glucose into cellular activity. When the cellular activity is not received glucose properly, then the glucose level will be higher in the blood tubes. So we can call it as a diabetic patient. So in order to avoid this process, our system is working. Whenever you consume some amount of food, it will be transferred as a glucose into the working tissue. The working tissue, it will taken as a foodstuff, and it will be converted as a two energy molecule. One is thermal energy, another one is chemical energy. Chemical energy can be utilized as a energy fuel for the working muscle, and thermal energy, we have to maintain proper temperature for your body, so that the other hormonal and chemical reaction will happen in a constant body temperature. So this is actually called glycolysis. Glycolysis, simply we can say, glucose can be transferred into the working muscle and converted as a energy sources for working muscle. Let us discuss step by step. So glycolysis as glucose's conversion into a fuel for a working muscle. It will be see, this is a blood. Here you can imagine a blood tube, and it will enter into cellular activity. Above or away from the mitochondria, it will be called as a glycolysis. So glycolysis, there will be a 18 stage. In this 18 stage, the first, 10 stages, the 5 stages energy investment phase, and another 5 phases energy conservation phase. After this, pyruvate can be transferred as a acetyl-CoA. Here 8 stages are there. So totally here 10 stage, here 8, and the electron transfer will be another 3 stages. So let us discuss. The glycolysis is the process in which glucose is broken down into produced energy for working muscle. At the end of process, you will get 2 energy molecule in 5 different category. That is called 2 pyruvate, 2 ATP, 2 NADH, and 2 water molecule. And at the end of process, these process can be carried out with oxygen, without oxygen, without any disturbance. So if presence of oxygen, it will be a citric acid cycle. If there is no oxygen, means it will be a lactic acid cycle. It will carry out. And this process taken place in the cytosol, in the cytoplasm. It is above the mitochondrial activity. So preparatory period, every preparation, every cycle we have to invest. Every business we have to invest something. Then only you can earn something. So in that concept, we have to spend something and get full return. So in that category, we have to invest 2 ATP molecule for this preparatory period, and the glucose can be converted as a glyceraldehyde 3-phosphate. In that we have to invest, and then after glyceraldehyde 3-phosphate, you will get return. So let us discuss about the preparatory period. What are the stages? In the investment phase, we have to spend 2 ATP molecule for glucose, and inclusion of phosphate, and finally there will be a 5 different phase, and finally it will be converted as a glyceraldehyde 3 phosphate. And at the end of calculation, we have to spend 2 ATP molecule to initiate the process. And at the end of 10th stage, it definitely you will get a lot of energy, that is its energy molecule per cycle. So in this first step, phosphorylation of glucose, it is nothing but glucose can be, or glucose added one more phosphate. For that, there will be a coenzyme hexokinase. So glucose can be converted as a glucose 6-phosphate. This is the process. Let us discuss. So one phosphate is contributed to initiate glycolysis process, and inclusion of one phosphate into glucose make it as a glucose 6-phosphate. So in this category, you can we can very well see hexokinase is the coenzyme where it will attach with glucose and attach with one phosphate and make it as a glucose 6-phosphate. So glucose can be converted as a glucose 6-phosphate. Here one phosphate, this phosphate can be attached with this sixth sixth base of carbon. So if it is attached here, it will be called as a glucose 6-phosphate. And one, the adenosine triphosphate converted released one phosphate and make it as a glucose 6-phosphate. Okay, then there is a adenosine diphosphate will be liberated at the, and there will be hydrogen ion is released in this category. So this is glucose. The ATP is given one phosphate here and make it as a glucose 6-phosphate. It is a first stage of glycolysis. For this, the magnesium has to involve, hexokinase has to involve and make it as a phosphorylation. Then only glucose can be converted as a glucose 6-phosphate. Okay. For this process, the glucose can be converted into glucose 6-phosphate. For this, hexokinase is involved. The phosphorylation phosphate group is added into 6-carbon molecule, and this process involves transfer of one phosphate from ATP into glucose and forming of glucose 6-phosphate. This is very very initiative phase. This is very crucial phase also. If it does not happen, definitely there will be no further reaction. In the presence of enzyme hexokinase and glucokinase only, it will be converted glucose into glucose 6-phosphate. This is first stage. Let us discuss about a second stage. This is rearrangement stage, where the glucose 6-phosphate can be converted as a glucose fructose 6-phosphate. So there is nothing changed. The base is going to change with the help of phosphoglucoisomerase. So this rearrangement carbonyl and hydroxyl group at C1 C2 stage in the bond. There will be a 6 base. In that 6 base C1 C2 is there is a change. For that change only, the glucose 6-phosphate can be converted as a fructose 6-phosphate, and which leads to reversible isomerization of aldose into ketose, or glucose 6-phosphate into fructose 6-phosphate. This is the second stage. The glucose can be converted as a fructose 6-phosphate. This is second stage with the help of phosphoglucoisomerase. Isomerase is nothing but changes the position and make it as a aldose into ketose group. So this is elaborate picturization, and here the phosphate, the glucose 6-phosphate, the changes will be happen in C1 C2. This is C1, this is C2. There is a change in this two area. That's why we call it as a glucose 6-phosphate. Here no, this is glucose 6-phosphate. Here the C1 C2 is changed its position, make it as a fructose 6-phosphate by glucose phosphatase isomerase coenzyme. In this stage also we need magnesium contribution. Then only there will be a possible of conversion glucose 6-phosphate into fructose 6-phosphate. And hexoisomerase catalyzed reaction with Mg, then only there will be a possibilities of changing the C1 C2 position, and another thing is the aldose converted into ketose group. Already I said. And then the isomerase plays important role to complete overall pathway of further glycolytic process. This is very important phase. And carbonyl hydroxyl group C1 C2 is a crucial step to carry forward the pathway of further. And third stage is very simple. Phosphofructokinase can be converted fructose 6-phosphate into fructose 1,6 biphosphate by adding one more phosphate group in the process. So where fructose 6-phosphate is converted into fructose 1,6 biphosphate, where one more phosphoryl group is transferred from fructose 6-phosphate and make it as a fructose 1,6 biphosphate by adding one phosphoryl group, I mean phosphate group. Here one more ATP we have to invest to make the process more energetic one. So this is also very important phase. And let us discuss this ATP. One more adenosine triphosphate we have to invest, and that one phosphate is combined attached with the phase one. So make it as a 2-phosphate energy compound. So that's why we call it as a fructose 1,6 biphosphate. For this process, we have to release one phosphate in this energy compound. So this step, second priming step of glycolysis, where fructose 1,6-phosphate is converted into 1,6 biphosphate by inclusion of one phosphate with the help of phosphofructokinase. So this step utilizes one phosphate donor from ATP by helping the enzyme phosphofructokinase and make it as a fructose 1,6 biphosphate. So next stage is fourth stage, aldolase. This is very important stage. Already we added two phosphate group. Those two phosphate group can be divided into two separate unit, and with 3 carbon, 3 carbon with one phosphate, 3 carbon with triphosphate. This process is carried out. In this step, fructose 1,6 biphosphate split into two different triose phosphate. Cleavage means divide from higher unit to lower unit or usable format. That is called glyceraldehyde 3-phosphate and dihydroxyoctone phosphate as two energy sources, different format. It is split from fructose 1,6 biphosphate. And the remaining step is the glycolysis is divided into two major area, where 3 carbon with 1 phosphate, 3 carbon with 3 carbon with triphosphate. This category is again it is divided, and it will carry out the further process of let us discuss and with the picturization. So here you can see one phosphate in the fifth and first stage. So this is first and this is fifth stage. There is a one phosphate unit. This phosphate unit can be divided into one separate unit, and second phosphate is divided into separate unit. First one is dihydroxyoctane phosphate, and second one is glyceraldehyde 3-phosphate. These two unit, one is more active. Glyceraldehyde 3-phosphate is more active than dihydroxy phosphate. This will not carry further process of glycolysis, but it will merge with the glyceraldehyde 3-phosphate with help of aldolase. Okay, this thing we are going to discuss. So in this step, fructose 1,6 biphosphate is split into two different triose phosphate, that is called glyceraldehyde 3-phosphate and dihydroxyoctane phosphate, and this process catalyzed by enzyme, it is called as a aldolase. The remaining step is glycolysis involve 3 carbon unit rather than 6 carbon. So previous stage there is a 6 carbon with a phosphate. Here 3 carbon, 1 phosphate or 2 phosphate, and another phosphate and 3 carbon. This will lead to glyceraldehyde 3 phosphate. It will carry further process. So after this, the energy investing, I mean the fusion of dihydroxyoctane phosphate, it will be merged with glyceraldehyde 3 phosphate. And so fifth phase, very simple phase, glyceraldehyde 3 phosphate can be readily degraded in the subsequent step of glycolysis, whereas dihydroxyphosphate cannot be readily available format, so that it will react, and hydroxy octane phosphate is isomerized into glyceraldehyde 3 phosphate. So again it will contribute, the dihydroxyoctane phosphate is contribute to promote glyceraldehyde 3 phosphate for further step. This is a energy investment phase. So energy investment phase, fifth phase is over. So now your the glycolytic process is getting ready to release energy releasing phase, where you can get ATP, GTP, and NADH, and water also released at the end of 10th stage. So in this phase, you can see the one phosphate, other carbon, it will combine with glyceraldehyde 3 phosphate, whereas dihydroxy phosphate 2 phosphate with the other carbon atom, it will form a 2 triose phosphate with help of triose phosphate isomerase. So this is the last stage of the energy investment phase. Triose phosphate isomerase is a very essential one. Then we'll see if this process is any disturbance, definitely there will be no energy conservation phase. So in the conclusion, the preparatory stage, there will be 1, 2, 3, 4, 5, 6 carbon with 1 phosphate is adding and make it as a glucose 6 phosphate. And this is second stage. And third stage, there will be a conversion of glucose into fructose, that is third stage. And fourth stage, again we have to add one more phosphate and make the chain is more more dense energy reservoir, fructose 1,6 diphosphate. This diphosphate again it will be divided into two category, that is dihydroxyoctane phosphate, and another one is glyceraldehyde 3 phosphate. So this 2 phosphate can be divided into two category with the 3 phosphate. So here afterwards the chain completely depends on 3 carbon with the phosphate. Let us discuss the further process, energy conservation phase, in the next video. So, and if you are very clear about this 5 stages, there will be no problem of glucose converting or anybody is affecting because of higher glucose in the blood. Pre meal and post meal also we have to be very careful, otherwise it will be more disturbance to your body and make it as more worse than any other diseases. Thank you.